Right. So for today, I want to talk about the cell cycle and actually the exact mechanism as to how the cell cycle proceeds. So how a cell actually moves from G1 to G2, or I'm sorry, from G1 to S to G2 to M phase mitosis, cell division. So the actual, molecular events that are occurring that drive that cell cycle progression. And it has to do with these protein regulatory molecules called cyclins, as well as these enzymes called CDKs, cyclin-dependent kinases. Okay, so let's take a look. All right, here we go. So the control of the cell cycle through cyclin proteins and CDKs. First, just in the early 1970s, there were some experiments that demonstrated the cell cycle is driven by these specific signaling molecules present in the cytoplasm, and that there was this rhythmic fluctuation. So, increased amounts and decreased amounts of these molecules that were actually controlling the cell cycle and the movement from the different the progression from the different phases of the cell cycle. So, as I mentioned, there's really two types of molecules, proteins, kinases and cyclins. So let's start with the kinases. As you can probably see, it ends in ASE, so we know it's an enzyme. It's actually a class of enzymes, kinases. They're not just involved in the cell cycle and regulating the cell cycle. Kinases are found in lots of other contexts as well. But what are they? So a kinase is an enzyme that is actually going to, so that activates or inactivates other proteins, other enzymes, but other proteins by, and this is kind of a funky word, but phosphorylating them. Phosphorylating them. So kinases initiate the transfer. So this is what phosphorylation and phosphorylating means. Phosphorylation is to add a phosphate group to the molecule, the protein. And so kinases actually initiate the transfer of a phosphate group. Oftentimes it's from ATP, actually. And they initiate they cause this reaction, catalyze it as an enzyme, of transferring that phosphate group to the protein in question or enzyme. And it's either going to activate that protein or inactivate it. So that's what a kinase will do. Okay, that's kinases. Now let's talk a little bit about kind of what these the cyclins are. And really, all you have to know about these cyclins are they're proteins, and they're going to fluctuate in their abundance. That's kind of where they get their name from. Cyclic, right? They're cyclically fluctuating in concentration in the cell. And so that's kind of what we mean by a cyclin, but they're proteins, right? So the key connecting thing sort of here are these so-called CDKs, cyclin-dependent kinases, and they're very much appropriately named as these types of kinases are only activated if a cyclin protein is bound to them. So a cyclin attaches to a CDK, and then the kinase can go ahead and phosphorylate other proteins. But without the cyclin attached to the CDK, so let's say, that there's no cyclins in the cell around, or it's very low concentration of these cyclin proteins, the CDKs are not activated. They're not going to go and phosphorylate proteins. Now, why does that matter? Let's take a look. I'm going to show you, well, we're going to talk a little bit about this specific example of why it matters. Probably the most famous cyclin CDK complex. So this is called MPF. It's most important, mostly because when MPF is active, and again, when you have all these cyclins produced in the cell binding to MPF, and I have a cool image of this, pretty well demonstrated on the next slide here. But once you get this accumulation of cyclins, they're going to bind to MPF molecules, these CDKs, and specifically, since it's mitosis M phase promoting factor, and this is the cyclin that's going to trigger and cause the cell to initiate mitosis and transition from G2 to M phase mitosis. And so I actually included I know it's kind of wordy, but it specifically, I wanted to include the how. And it's kind of some neat research here about how this activated kinase, this MPF CDK, how is it causing the cell to progress and start dividing mitotically? Well, it actually is going to phosphorylate proteins of the nuclear lamina. The lamina is these structural cytoskeletal proteins. Intermediate filaments are kind of a subcategory of cytoskeletal elements. And the lamina borders the membrane, the inner membrane of the nucleus. And actually, once you add a bunch of phosphate groups to the nuclear lamina, it actually causes it to start to break apart. And as we know, we might not have talked about it yet. It depends on the class. But one of the first key events of mitosis is the breakdown and the dissolving of the nuclear membrane. And so that's kind of a very exact, specific way in which the cell is going to cause this progression and initiation of mitosis, and prophase being the first phase. And again, in prophase, one of the key events is this dissolution of the nuclear membrane, and MPF is phosphorylating the structural component of the membrane, or at least part of it. So I thought that was kind of an interesting example to share. And then, of course, this is just the classic, one of the most important examples, but other checkpoints like the G1 checkpoint is going to be regulated by cyclins and their CDK partners. And so here's a really nice image demonstrating this kind of mechanism and sequence of events. And so as you can see, there's this on the right side, the cyclin accumulation. So as the cell starts to produce more cyclins, more CDKs become activated, such as MPF, which is actually just, it's going to cause this progression through the cell cycle. As CDKs that are activated by cyclins start to phosphorylate proteins involved in cell cycle progression from S to G2, et cetera, et cetera. And then, of course, the cell cycle actually will start to slow down and not progress and go quickly through it as the cyclins become degraded by other enzymes in the cell. But the cyclins will break down, they'll degrade, and so the CDKs are inactive. And so really the key point, the key takeaway here is that the cell cycle is regulated and driven primarily by the presence or absence, right, of cyclins that are activating and binding to these CDK enzymes. I think that's it. Yeah. So again, it's this rhythmic activity or fluctuation rather in concentration of cyclins that is causing the cell cycle to move or to not progress and not move. And that's really it. All right. Cool.